Hi, my name is Tony Callowson, Head of Communications and Engagement at Cambridge University Health Partners, and I'd like to welcome you to today's Cambridge Biomedical Campus on virtual tour event. Since we moved our talks online, it has been fantastic to have so many of you join us to hear from the amazing clinicians, researchers, and academics which we have in Cambridge. And you can watch all of these events all again on the CBC YouTube channel. This lunchtime, I have the privilege of introducing consultant neurosurgeons Mr. Stephen Price and Mr. Richard Mayer, co-leads of the Brain Cancer Virtual Institute at the Cancer Research UK Cambridge Centre, who will be discussing the pioneering research in Cambridge that's aiming to improve outcomes for brain tumour patients. Stephen is an NIHR clinical scientist at the University of Cambridge and honorary consultant neurosurgeon at Addenbrooke's Hospital, while Richard is assistant professor in neurosurgical oncology at the University of Cambridge and an honorary consultant neurosurgeon at Addenbrooke's Hospital. Hopefully I've got all their job titles right. As usual, throughout the talk, if you're watching through Zoom, you can ask questions at any time using the Q&A box, which will be addressed at the end of the talk. And we'll do our best to include any comments made by those on YouTube Live, but please don't be too disappointed if yours is not included, as we often have a very chatty audience. And finally, to reassure you all, you won't be seen or heard throughout this event, but it will be recorded to be accessed on our website and the CBC YouTube channel in the future. So without any further delay, let me hand over to Stephen and Richard.
So thank you for that introduction. I'm Stephen Price, as you heard. I'm a consultant neurosurgeon, and I'm going to make a start and discuss some of the work that we're doing to help improve the outcome for patients with brain tumours. And my colleague Richard Mayer will then take over later on to discuss some other aspects and some of the really exciting work that's happening. So what I'm going to do is outline a bit of the problem, how we might improve prognosis for these patients, and then Richard's going to talk a bit more about some of the new things that are happening, particularly around the Precision Brain Tumour Programme and other trials and other data sets that we are hoping to work on, and give you an idea of our vision for the path forwards. So brain tumours, just as a bit of a background, there are lots of them, 235 different types of brain tumours. The commonest are benign tumours that come from the lining of the skull, so-called meningiomas. Tumours that spread from other cancers, particularly lung and breast and melanoma that spread to the brain, so-called brain metastases. And the ones that we're going to focus on here are the malignant tumours of the brain. They are the gliomas, and I'll talk a bit about those in a minute. Now, these tumours are relatively uncommon, but still they carry a large burden of disease. They're the commonest cause of cancer death in children. They're the commonest cause of cancer death in young people, so that's people under the age of 40. And if you take an individual person and work out how many years of life they lose, on average, you lose more average years of life with these tumours compared to other cancers. So for the individual, these have a really big impact. And one of the big things that's different about brain tumours compared to other cancers is that it robs us of what makes us individuals. It takes away some of those functions that help us. And I'm going to touch a bit on that a bit later on. So let's just start talking a bit about gliomas. What is a glioma? Well, the gliomas come from the glial cells, and these are the cells that are within the brain that support the neurons that do the main work of the brain. And you can get some of these tumours that come from these supporting cells that support the neurons, called the oligodendrocytes, and they become oligodendrogliomas. Others come from the astrocytes, and who are other supporting cells, and they can become either an astrocytoma, or if they have, and that's usually associated with a mutation called an IDH mutation. If they do not have this mutation, then they become what's referred to as a glioblastoma. And unfortunately, 75% of all gliomas turn out to be this glioblastoma. And you can see the difference here in the different survivals between these different tumour types. And 14.6 months of average survival for patients with glioblastomas is for that group of patients that get the most treatment and are treated, have the most intense treatment. And a lot of patients are not fit enough for that. So what do we do with these patients at the moment, these glioblastoma patients? Well, the first thing is we try to take away as much of the tumour as we possibly can without causing problems. Patients then go for radiotherapy with some chemotherapy, and that's been shown some years ago that this has an improvement in outcome for patients. Now, if you look at other cancers, they've made some big improvements by adding in targeted therapies and adding also in therapies that help the immune system kill off the cancer cells. But unfortunately, these have really not translated very well into this group of patients, and we don't have any new treatments from that. And so what happens is we take these tumors out. So here's the end after an operation. So there's a little bit of tumor. We can just see a tiny bit there that's left behind after surgery. And these patients will then go on to have radiotherapy, and you can see the tumor comes back. And the tumor comes back usually in exactly the place where we left it. And in most patients, it comes back within the year. And what is really depressing is that if you look at this report that came out a few years ago, although there have been lots of drugs registered with the European Medicine Agency for treatment for things like lung cancers or blood cancers or breast cancer, there have been no new drugs registered for the brain. And at the moment, there is no new treatments out there, and we have had no change in our treatment since that paper that looked at the radiotherapy and chemotherapy back in 2005. And our improvement in outcomes have really stayed static for the last 20 years. So how might we improve things? Well, the first thing, which is really clear, and it's coming out from breast cancer, is that this is not a single disease, but lots of different disease. And can we characterize this? And this is something that we'll talk a little bit about later on. How can we improve current treatments? And I'll talk about how we might improve surgery in a bit. We need better clinical trials that allow us to explore new treatments, new therapies, but also combinations of new therapies, which I think is going to be the key thing here. Also have the ability to look at big data sets to explore rare patient subgroups, so very small groups of patients who may have a very good response to certain treatments. And then be able to integrate our scientific and our clinical resources well. So as part of this, our vision is the CRK Minderoo Brain Cancer Virtual Institute, which I'll explain in a second, the Precision Brain Tumor Program, the 5G platform trial, and the Universal Cancer Data Bank, as well as some of the international work we're doing. My colleague Richard Mayer will discuss in a bit. So this was the CRK Minderoo Brain Cancer Institute was launched earlier this year. The aim is to improve the treatment in front of us, but also to improve the treatments for patients in the future. We are very worried the fact that a lot of new treatments that come out can take quite some years before they ever get to patients. And we're very keen to find how we can improve things for patients today. And this is the structure of our program. So we are aiming to ask clinically important questions that require scientific answers. So we have a series of programs, and on this picture here, you'll see around the outside of it, these are different areas that we think are really important, and also that we have got some experience within the whole of Cambridge, whole of the university to manage. So imaging and tumor metabolism is a big area for us. Developmental neurobiology. How does the processes that are involved with the brain development go wrong and cause cancers? That's mostly looking at cancers in children. Heterogeneity, recurrence, transformation, and treatment resistance. Why is it that we find these tumors don't respond to treatments? And even when they do respond to treatments, why do they suddenly stop responding to them? And how can we overcome some mechanisms for that? And then the big area that has had little looked at the moment is looking at how we might better manage patients with metastatic brain disease. In other words, tumors that have come from other cancer sites and have gone to the brain, because they often fall between the gaps between research going into brain tumors and research going into other cancer areas. And supporting that is our Icarus, which is our biorepository. And the idea is we want all of this information to feed into our MDT, which is our decision-making tool we use when we discuss new patients between a variety of clinicians to talk about what's the best way of treating them. So we want to provide this information for that. And Richard's going to talk a bit more about that at the moment. And here's the team. Richard and myself are the two co-directors of this, with Jill, who is our program manager. And the program itself has quite a number of members involved with it, from clinical members through to members involved with very scientific backgrounds. And you can see on the other side here the departments these people have come from. So clinical neuroscience and oncology, obviously the two big ones, but we've got members of the program that are involved with a whole variety of other things, from engineering to the Cambridge Stem Cell Institute, from molecular biology through to psychiatry, surgery, biochemistry, pharmacology, and even the Department of Chemistry. So it's a real big mixture of different organizations that are helping with these problems. And I mentioned we wanted to ask clinically important questions that we could find scientific ways of approaching. And this is just a series of the questions that we've put together for each of these topic areas. And I'm just to sort of explain the sort of our approach to this, I'm going to take the top one. How can we use imaging to optimize brain tumor surgery? And the problem we have for surgeons who spend our time operating on these malignant tumors is that we have to walk what I refer to as a neurosurgical tightrope. So we have a tumor. If we take out part of it but leave a lot of the tumor behind, then all that happens is that tumor grows back very quickly, and the patient will then have the problems of a tumor growing back and succumbing from that. Unlike our colleagues, for example, who are operating on the bowel, where they can take out a larger area of the bowel, we can't do that. If we go too far, this is an example of us going too far into the brain, we can damage normal brain structures that can have an effect. And we know that the best outcome for our patients and the best survival for our patients is to take away as much of the tumor as we possibly can, but without causing them any problems. So what is our target? What are we going to take out? Well, when we do an MRI scan, and this is an MRI scan example of a patient, and what we get with it is we get a series of different sequences that tell us just slightly different bits of information. So this is when we give an injection of the contrast agent, and you can see the tumor lights up. All this white area here is lit up with tumor. And we think about that as an area that is full of tumor. And our primary aim is to get rid of this part of the tumor as much as we can. This is another image we see, and this is an area of what we call non-enhancing tumor. And we think about this as a bit of tumor invasion, and we've been brought up to think of it as a bit of swelling around the brain as well. Actually, what we now know is that this is full of tumor. But to remove all of this would almost removing a quarter of this person's brain. It would be impossible for us to do that. So what we're trying to do is use advanced imaging techniques to understand this and to then plan things and try to work things out and plan the best way of removing these tumors. So one of the techniques we're using, and I've been very interested in the last few years, is called Diffusion Tensor Mri, and basically works like this. If you think of the brain, it consists of areas of the cortex that are the areas that do some of the processing. And there are these tracts or white matter tracts that are like the cables within the brain that link it. And we have imaging methods that is sensitive to the movement of these water molecules along these cables. It's a very directional flow of water. Now, if you then disturb that by putting a tumor in, you then find you lose that directionality, you lose that ability for water to flow in one direction. And that's what we use as part of our imaging techniques. And we can use other imaging markers as well that tell us, again, different things about the tumor and give us different things about the pathology and what's happening within those areas. And this is an example. This is going back quite some years. This is a patient who had previously had an operation on this side of the brain and then presented with a tumor on the other side of the brain. And we wanted to know why didn't it occur locally and what was the mechanism of spread from it. And this is the pathway that separates the two halves of the brain, that link the two parts of the brain. It's called the Corpus Callosum here. And it looks absolutely fine. We can't see anything wrong with it. But if you look here where the arrows are, we could see with our technique a much more sensitive method to detect these changes. And then what we then applied was a new technique and developed new techniques of being able to assess this. And this technique that we used actually comes from engineering. It comes from geomechanics. So this is how engineers describe the stress strain patterns within the soil around skyscrapers and around buildings. And we use exactly the same technique. And we've separated into two components. So this is the Q component here, which is the central one. And when we took biopsies of that, what we found was it was pure tumor. And then outside of that, we have this area called the P area, and that has got tumor cells that is growing into the normal brain. And interestingly, when we took biopsies outside of the P area, we couldn't see tumor at all. And what we've done is we've looked at this over the years and tried to understand what happens when we remove this. And what we've done is looked at how much of this we can remove. So this shows a survival curve. This is the number of people that are still alive. This is the number of time. And the idea is obviously you want to stay alive as long as possible. And you can see that this dotted line here is doing better than this other line here. And this area with the dotted line is where we removed most of this tumor. And we removed much of this Q area, the area we said was full of tumor. And it seems this might be a better marker for understanding, for planning how we should be removing these tumors in these patients. The problem, as I said, is you've got to be careful of the normal brain. And although we are quite good at monitoring things like motor function and language function, which we can do with patients awake talking to us during an operation, what we're not very good at is other things, including cognition. And this is a big survey done some years ago that looked at what were the problems that patients with brain tumors faced. And one of the big things is they have these what's so-called cognitive problems. They can't remember things. They have personality problems. They have memory problems, and they struggle to communicate. And this is a big issue. But how do you measure these? Well, we used a technique. We've developed a technique with assistance from the Cognitive Brain Sciences unit of using a battery, a tablet-based app that allows us to test these functions in patients. So it's easy to administer. You can do it. My colleague who does a lot of these refers to it as very NHS proof. He does it in cupboards and all sorts of things with patients. It's a very small area you need to do it. Patients prefer it to the paper tests, and it is something that they can do usually very easily within about half an hour, 40 minutes. And what we did was when we looked at this data, we found that we were detecting a lot of problems. So the ones in pink here are the glioblastoma patients, and pretty much every patient had at least one deficit we picked up. And there were multiple ones in individual patients. Now, one area we're particularly interested is this, emotional recognition. Now, this is the ability to tell how someone feels just by looking at them. And this is important, part of what's called social cognition. It's how we interact with others. Because again, from this brain tumor survey, we know that inability to interact with others leads to patients feeling awkward in social situations, and they withdraw from it and become very isolated because of this. So what we do is we give patients a series of faces to look at, and we say and ask them to point on the screen and touch the screen to say what they think the emotion this person is showing. And we compare that to normal individuals. We've got a lot of data now on these normal individuals, what we think is normal. What we wanted to know is which parts of the brain are involved with this. If we know which parts of the brain are involved with these areas, could we avoid damaging them with surgery? If we knew, because we know the areas are involved with some of the motor functions, for example, or language functions, and we know how to avoid them, could we do the same with this? So we looked at the parts of the brain that were changed in these patients that had emotional recognition problems. So these areas here you see in red, and we compared them to a whole list of other functions that were very similar to do with looking at things and recognizing things, but they only were found in these patients that had problems with emotional recognition. And we found there were a certain number of tracts on the right side of the brain that were dealing with it. Now, these tracts are identical to what we see in patients who've got head injuries with the same problems, which suggests it may be through the similar sort of mechanisms. And it may mean that we can use rehabilitation tools that has already been developed for these patients to improve outcomes in our patients. But the second thing we noticed, which was very surprising, was this happened even in patients who had left-sided tumors. In other words, the idea that we're taught in medical school that we have a bit of cortex and it's connected to one mass tract may not be completely correct. And what we're looking now at is, again, with work we're doing with the Department of Psychiatry, is actually looking at networks, how the brain functions as not just a small area working on its own, but has a network and how it works together. So here's an example of a patient with a tumor. And we think there's networks which are all connected by these what's so-called hubs, these sort of circles you see here, and they all connect together. And the ones in red are the ones that are just by the tumor here. And the size of them depends on how many connections they have. And what we're able to do, and we're starting to do now, is trying to sort of model it. So if we take out some of these here, we can then work out by a series of measures how well this network is going to work and try to work out, can we predict patients who we're going to make worse by these treatments? So this is an example of one of the workflows we're going to do. And this has led to a study that is currently ongoing, that we are looking at how much of the tumor, what we should be removing for the tumor, in other words, what is the true extent of it, but also what can't we remove, what will we cause major problems for patients, particularly with cognitive patients. And the idea is we're coming up with an individualized treatment volume that we are then going to take back into the operating theatre and allow us to change and improve the way we do our operating. And the initial data we have suggests that this is leading to some improvements in survival by the more of these areas we take out. But by doing what we normally do, but just doing it a bit better. Okay, I'm going to leave it there. I'm going to let my colleague now carry on and talk a bit about some of the molecular oncology that's going on in Cambridge.
Thanks very much, Stephen. So I'm just going to share my screen. Let's see if I can get the right screen. Okay, so I'm going to talk, as Stephen said, I'm going to talk about some of the molecular oncology aspects of what we're doing here in Cambridge to try and develop brain cancer care into the next generation of treatments. So going back to that initial diagram that Stephen sort of alluded to, where we had the sort of target-based description of our program, essentially the multidisciplinary team, which is this MDT, is where all patients who have a brain cancer putative diagnosis are referred to, and from where we discuss all the patients post-operatively to produce treatment plans for them. So what we wanted to do is make that MDT, sort of revamp it, if you like, as the center of our model for brain cancer care. And this is a description of how we're going to do it. So what we can see there is MDT in the middle. The Icarus Biobank and the Advanced Cancer Imaging programs lead from it. And what that means is for every patient that comes through the MDT, when they go for surgery or when they need further imaging, that's discussed centrally, and we can plan exactly what it is we're going to do. So through that Icarus Biorepository, which is essentially where we take samples from patients at surgery, obviously with their consent, and bank them, we can do various things. So we can enroll them in the Precision Brain Tumor program, which I'll talk a bit about in a minute, whereby we get whole genome and whole transcriptome sequencing. So we look at the changes that have happened within the cancer cells that have caused them to become cancerous. We also enroll them in the liquid biopsy program, where we take samples of blood and urine and sometimes cerebrospinal fluid to look for the changes ongoing in the cancer. And we also take cultures of the tumor, so we can grow those tumors in plastic, if you like. And what that does is it gives us a method by which we can affect a change in those cancer cells and see what they're going to respond like. The Advanced Cancer Imaging program is using more molecularly targeted imaging-based techniques to try and explore more fundamentally what's going on within the cancer at the functional level. I'm not going to talk about that much today, but the important thing to say is that those programs don't happen in isolation. The key thing with our new sort of revamped program is that we then feed them all back into the MDT, and we do that via separate meetings, specifically the Genomics Tumor Advisory Board meeting, which is the GTAB that I chair. So that means that the information we get back from the Precision Brain Tumor program, for example, can be analysed offline and brought back to the MDT. So then we can stratify patients into our new clinical trials platform, which is called 5G, which I'll talk about later, but also into other platforms such as RadNet to understand a bit more about radiation and radiotherapy. So this is a really exciting new program that we've launched in Cambridge at the start of the year that I think brings everything together towards a cogent goal of really improving outcomes, not just for the patients in 10 years' time, but for the patients in front of us. So I mentioned earlier, Icarus is our biolab, and essentially what we do here is preoperatively we consent patients for entry into this, and what it means is we can store frozen samples, we can store samples that we can culture, and we can take these liquid biopsies. And it's from this platform, from this laboratory, that we can perform our own experiments, but we can also share tissue, obviously with the patient's consent, with other researchers throughout Cambridge and internationally, to really provide the tools and the samples that are necessary to start looking at the next generation of therapies and next generation of investigative tools to really improve those prognoses moving forwards. We've been very successful. We've now got well over 400 patients, and we're over 1,000 biosamples now. And this is just in the last 3 years. And this is something that we're really, really proud of, and has been really, really useful across a number of projects, not just in Cambridge, but also nationally. The liquid biopsy program is something that I'll talk to you in a bit more detail about. Essentially, what a liquid biopsy is the changes that we see ongoing in a cancer are not just located within that cancer. So what we see is not only a systemic response, but also some of the changes that we see, for example, in the DNA or the RNA, so the stepping stone to protein within a cancer, are released from that cancer into the circulation, so into the blood, and then out of the body through the urine. And so what we've tried to do is harness that, and in conjunction with various collaborators, we're looking at whether we can use patients' blood samples, patients' urine samples, and patients' cerebrospinal fluid samples to get a better understanding of their cancer. And in order to do that, or the reason for doing that, is various. One thing, we could potentially look at using it to diagnose patients without having to do a biopsy, for monitoring patients' disease to seeing whether it's coming back, rather than having to do the serial imaging that we do at the moment. But also looking to see whether we can target and plan treatments based on the information that we get from this blood test or this urine test. So it's really very sort of science fiction-y almost, but we are showing that we have some interesting response and some results. And what we see there on the right are some of the papers that we've published just out of Cambridge alone over the past 3 or 4 years, showing that there is some promise in this technique. Now, when we started out doing this, what we found is that actually we couldn't have picked a harder tumor if we tried. Across all of the various cancers in which liquid biopsy has been thought about, glioma, so the primary malignant brain cancer, has been the most difficult traditionally to look at. So what that meant is that we had to be quite creative in what it was we were looking for within the blood. And actually the amount of DNA that's come specifically from that cancer is quite low. However, what we were able to do is look sort of across that cancer and across the blood and say, actually, what's changed in the blood of a cancer patient that's different to a patient without cancer. And we were able to find various metrics, such as how DNA within the blood is fragmented, so how it's broken up by the enzymes within the blood, and that will tell us whether that patient potentially has cancer or not. We were able to look at the cerebrospinal fluid, which is the fluid that bathed the brain, which we access via a lumbar puncture. And we were able to show, just with a fairly cheap sort of 30 to 50 pound test, looking at that cerebrospinal fluid, we could see changes within it that were indicative of the presence of a cancer. So we're not biopsying the cancer, we're doing a lumbar puncture, but we can see those changes in the cerebrospinal fluid. Now, obviously, taking cerebrospinal fluid, if any of you have had a lumbar puncture, it's not a very nice procedure. So this isn't something I think that's clinically going to translate, but it means that we can use similar approaches by looking at things like the fragmentation pattern of the DNA that's in the circulation, but also the copy number, so the amount of copies of each gene from the cancer or from the normal in the circulation. And again, we can use these techniques to detect cancer in patients. What we did for this study was we compared the cerebrospinal fluid, the plasma, and the urine. And what I mean by plasma is the blood once we've spun all the cells out of it. And what we're able to show here in figure E, it's quite a busy slide, I do apologize. But although the fraction of DNA that was informative is higher from the cerebrospinal fluid, actually we were still able to detect a good signal across plasma and urine samples. So there is signal there. I think what we need to do is actually just look at better ways of understanding how to detect it and how to look at it. And these are some of the methods that we used. We used a technique called machine learning, which is essentially we were able to pick out different factors and different ways of analysing the DNA fragments that were sitting within the plasma or the urine. And by looking at those in combination, we were able to boost our sensitivity, so our ability to identify abnormal bits of DNA in the blood and the urine in order to sort of refine our process. So I'm next going to talk about the Precision Brain Tumour Programme, which is something that we have started since the beginning of the year in collaboration with Minderoo, who are an Australian charity. Essentially, the idea of this was to perform whole genome and whole transcriptome sequencing. And what I mean by that is taking bits of a patient's cancer at operation, extracting the DNA, which is the genetic code, if you like, of a patient, and in which we can identify the changes that have likely led to the cancer, but also looking at the next step of transcription, which is RNA, which is then the building block to protein, and combining those two analyses to try and understand whether we can better diagnose patients, but also stratify them into clinical trials. So the aims of our program were really, first of all, just to see whether we could do this. Traditionally, sequencing is something that's taken many months and has been very expensive. And what we wanted to do was to see whether we could bring that down to under 3 weeks, and whether patients wanted us to do this, whether it was something that they were up for. Secondly, we wanted to see whether it was useful. It's all well and good saying we're going to do these sort of highfalutin scientific techniques, but are they actually going to give you any benefit in the clinical sense? So we wanted to see how it affected diagnosis, whether we were able to prognosticate, and also whether we were able to target therapy based on our data. Once we've done that, the idea is then to democratize this around the country. This is not something that we just want for patients in Cambridge, as it currently is, but I think it's better to use the carrot rather than the stick, in terms of if we can show it has benefit, perhaps people will pick it up, and we can provide the exemplar and help them do that. So our experience so far in the first 8 months, we've now actually recruited over 80 patients, which is fantastic. 100% of patients approached to be in the study have agreed to be in it, which is, again, I think, a good reflection of people's desire to be part of scientific studies, but also to have their genome sequenced. We've codified the new tissue pathway, which was no trivial task, very difficult. Hospitals are complex ecosystems, and science is equally complex. But we've managed to get that pathway up and running. And we have the new Genomics Tumor Advisory Board, which I chair, which meets weekly to discuss the patients that have gone through the program. What's really been exciting is 20% of the patients with glioblastoma that have gone through our pathway, we've found targetable mutations where we can potentially stratify them into new precision therapy trials. And that's more than we thought we would get, but really fantastically encouraging for this approach. So this is our pathway. Essentially from surgery, we have the sample, very small sample that's taken, which obviously would have been taken anyway during the operation. It goes through the Icarus biorepository to our Cancer Molecular Diagnostics Laboratory at the University of Cambridge via my research assistant, Gemma. We then send that off to Illumina, who do the DNA and the RNA sequencing, and then it's brought back to the GTAB, where our amazing clinical scientists, led by Patrick Tarpey, will analyze the genomics output. We can then discuss it sort of offline, if you like, at the GTAB, and then feed back the salient findings to the very busy Neuro-Oncology MDT, where we can stratify patients as required. And the idea is that we do all this within 3 weeks. So this is the overview of our first 8 months. What we found, as I said, 79 patients. We're projected to get 120 patients in our first year, which again is fantastic, because we'd initially said we were going to get 50. So we've really over-recruited, which is a brilliant exemplar of what this sort of approach can do. Our median surgery to sequencing return time is 17 days. So again, that's really fantastic. We have had a couple of outliers, but that's been for logistic reasons. What do we get from this program? Well, these are just some of the basic outputs that we have. So we can see 2 different cancers here. What we have is what's called a copy number plot. So essentially, the chromosomes of the human body are mapped out along the bottom, and what we can see in the top picture is those blue lines that are circled in red. And what that means is there's a underrepresentation of that part of those 2 chromosomes in our sequencing run. And this is indicative of a specific type of tumor called an oligodendroglioma. Moreover, in the bottom, what we can see in the red line that's circled there and the blue line that's circled is an amplification or an overrepresentation of part of chromosome 7 and a reduced representation of chromosome 10, which is again indicative of a glioblastoma. So this is something just to reinforce that what we're doing is appropriate, and we are seeing these changes. But these changes actually do help us. So we have an example here of a patient who had a tumor that had been graded as being low grade by the pathologist. And actually what we found was that they had these changes on their genome that were indicative of perhaps a higher grade disease. So we were able to go back to the pathologist and say, right, can we take a few more blocks and have another look? And indeed, by doing that, we found that the tumor was indeed high grade. So although for the patient, it wasn't fantastic news, obviously what we need to do is make sure we get the most accurate diagnosis to them so they can be treated appropriately. Moreover, as well as those copy number plots, what we also see is specific mutations within specific genes that we can read back and say, actually, we can find these, and these are all potentially targetable with current therapies that are available. And so we fed these back to the MDT and to the various oncologists involved. So these patients have the opportunity to be enrolled in clinical trials, whereas previously they perhaps wouldn't with standard of care histology. And as I say, we have about 20% of patients that we have made this available for. So just briefly, why do we want to personalize therapy and what does it mean? Well, essentially, currently, we adopt an approach whereby conventional glioblastoma therapy involves taking a group of patients with a histological diagnosis like glioblastoma, giving them a standard chemotherapeutic drug. Some people will respond, some people won't. But there's very little option as to whether we can give them anything else. The idea with personalization is we stratify patients depending upon the specific mutations that we find within the cancer to different treatments. And those treatments have a scientific rationale to be effective against the change that we see in that patient's sequencing data. And actually, if we go back and we look at the literature on this type of approach over the past 10 years, we find that the nihilism that perhaps exists within the oncology community towards brain cancer personalized therapy is perhaps a little bit unfounded. And actually, a recent systematic review done by one of my students showing that actually in up to 62% of patients, there may have been some benefit. It's just that perhaps the reporting outcomes from these trials weren't as effective as they might be. We know that glioblastoma is made up largely of 3 aberrant pathways. So these are intracellular signaling pathways that have essentially become dysregulated, okay, through these amplifications that we saw in those copy number plots, or through the mutations in genes affected during this pathway. The cells proliferate uncontrollably, survive when they perhaps shouldn't. And so the idea is if we can identify these, we can block these specific pathways. We have a group of medications that we can use. And actually, if we look at the various drugs that are available to us, and if we look back through the precision data from these last 80 patients, actually there are multiple potentially targetable mutations already present within these samples that we aren't targeting. So what we did in combination, or in collaboration rather, with the Royal Marsden Hospital is set up this 5G trial. And so it's run by Dr. Juanita Lopez, who's a medical oncologist from the ICR, so the Royal Marsden down in London. And the idea is to provide a platform trial so that when we identify new medications that are potentially suitable, we can use the Mindry Precision Brain Tumor Program to target those medications. So we can identify the genes that have gone wrong. We've got the platform ready. When we get a new drug in, we can add it on like a Lego block onto the platform and stratify patients accordingly. Now, in order for drugs to get on this platform, obviously they have to be safe. Ideally, they have a dose optimization program already, but we can build that in. And the idea is that we're testing a proof of concept. Now, we're not expecting these to be miracle cures. And indeed, we expect them, a large number of them to fail, if not all of them to fail at some point. But I think the information we can glean even from the people who respond for 4, 5 months, 6 months, a year will be very useful. And we can even go back into those patients potentially at recurrence, perform further surgery, analyze what's changed, what's gone wrong on a cellular level, and see if we can think about combination therapies that might improve outcome further. So I think that the challenges associated with glioblastoma are sort of, I wouldn't say we're circumventing them, but we're starting to move past them. I think initially there was limited interest from the scientific community. I think there was a degree of therapeutic nihilism. You know, what's the point? And I think that what Stephen and I have seen certainly over the last 5 years is that's changing, fortunately so. And it's now been registered as a cancer of unmet need with CRUK. Few targeted medications have been trialled, and when they have been trialled, they've largely been trialled in untargeted populations, which is as silly as it sounds. But for logistic and pharmaceutical reasons, that's been the case. And what we're trying to do, certainly with the 5G trial, is get around that. We do want to integrate large and diverse data sets, and I think data is power. And that's something that we're looking at doing nationally and also internationally. And through the program, we're looking to connect these diverse scientific and clinical disciplines for this shared goal of improving outcomes for patients with primary brain cancer. So I think moving forwards, the CRUK Minjury Partnership is a paradigm shift for our approach to this disease. And by collaborating with the Tessagel Brain Cancer Mission, we hope to provide additional benefit and opportunity. This isn't something we want to keep for Cambridge. We obviously want to democratise this, to federalise this. It's very difficult to try and do these things at scale to start with. I think, our understanding or the best approach we feel is to try and do it locally to start with, to show that it works, and then to really ramp up when that's the case. So it's much more carrot rather than stick. I think that's about time. Just obviously, this isn't work that Stephen and I do in isolation. Both of us have large lab groups and large clinical and scientific collaborating networks that we work in that are far too numerous to document here. But a big thank you to them, and obviously a big thank you to all of our funders, without which none of this work would be possible. And then finally, and not least, thank you to all our patients and their relatives for the amazing help that they give us in this research, and the samples they provide, the time they provide in being involved in these research studies. So thank you to you all.
Lovely, thank you ever so much, Stephen and Richard. As always, we thought that these talks, they are incredibly fascinating, and to learn about the challenges that you face and how you're overcoming them. It's great. So what we will, I've got a few questions that have been sent in, but if anybody wants to ask any specific questions, if you just add them to the Q&A, that'd be fantastic. So I guess kind of almost a general one that have come through. Someone's asked, will genetic testing for newly diagnosed brain tumour patients be available at other hospitals soon? I think that's sort of something you sort of started to talk about, Richard, about how that process of scaling up goes, because I know that a lot of the time people talk about the research that Cambridge does, which is fantastic, but what's that process of getting it out to the rest of the UK?
Sure. So the NHS have actually now commissioned whole genome sequencing for all patients with primary brain cancer. So the NHS have actually now stumped up the money to do that. The problem is it's not just about getting the sample sequence. What you have to do, and what I sort of tried to show with our pathway, is you need the whole pathway there. So you need the sample to be taken, to be stored appropriately, for the DNA to then be extracted, for that DNA to be in the right place, for that sequencing to be done, for then it to be returned, to then it to be interpreted, and then to be fed back in. So it's not just about the sequencing which has been commissioned, it's about the whole pathway. So that's what we've managed to set up here in Cambridge, which has taken us a few months to do so, and we're hopeful that we can provide the exemplar. There are other programs out there like Brain Matrix, which are also trying to do the same thing. They're trying to do it more sort of across multiple sites, which obviously, as I said, is a greater challenge, but I think will also be very useful in getting this rolled out. I think the more we can do to show that it's beneficial, the easier it will be. And again, that's why what we wanted to do locally was to demonstrate that it had efficacy, not just in diagnosis, but also in treatment planning. Again, just to provide that carrot for trusts whereby they have a busy clinical service, and this perhaps isn't top of their priority list. If we can demonstrate that not only is it available, but it's useful, I think the money to get that pathway set up everywhere will be more forthcoming. Thanks, Richard. Was there anything you want to add, Steve?
No, I completely agree. I think this is true of all technologies in the health service, is that although they can tell you some nice information, which is great, unless you can do the work that we're trying to do that shows that this is making a difference to patients, and you can use that information to change how you and improve outcomes for patients, then it's very hard to get it taken out.
Thank you. Someone's asked one of the questions, can you talk more about the collaboration with Royal Marsden? Is there not the capability to do that kind of work in Cambridge, or can you talk more about the benefit of collaborating further afield? I suspect that this is one of the questions I was going to ask, whether the new Cambridge Cancer Hospital how that would help you to do your work, but I think it's easier to answer this question about Royal Marsden first.
I mean, I think that collaboration is a great thing to do, and I think it really, provides benefit, so you become more than just a sum of your parts. I think in neuro-oncology, traditionally, the post-surgical treatment has largely been directed by clinicians that are radiation oncologists, so who give the radiotherapy. And consequently, there's been fewer medical oncologists who are a different sort of training to the radiation oncologists. And they're the ones that traditionally deal with the precision therapies. So within brain, there are very few medical oncologists working in our country. And the Royal Marsden have one of them. And that's really as the simple reason why we got this collaboration up and running. And I think that not only that, Juanita is a fantastic early phase clinical trialist and is very interested in this space. So it was sort of serendipitous, but also slightly engineered in that sense. I think that, the technology isn't necessarily, mutually exclusive. We can all do the sequencing, but as I say, it's sort of the sharing of knowledge and experience and sort of the desire to move this forwards, for the patients, that I think it's tapping into the pockets of that around the country, and that's what we've done with this collaboration.
You said that there are a limited number of you in the UK. Is that repeated across the rest of the world, in America and in Europe and in Asia as well?
I'm not sure exactly to the extent that it is in the UK. I think largely, traditionally within brain, treatment has been performed by the radiation oncologist purely because everyone has radiotherapy as the sort of mainstay of treatment. But it is changing, and there are, we do collaborate internationally now. We collaborate with UCSF, with the NIH in Washington, DC, and they have medical oncologists there very much interested in the brain space. And so that's something that we're actively pursuing. At the same time, we're trying to change the narrative here in the UK, and we're sort of saying, not just to the powers that be, but to trainees coming through, brain is open for business, if you like, and we're really interested, if you're interested, in having those discussions and seeing what we can do to facilitate your training to make sure that you have what you need to become a medical oncologist in brain cancer.
Thanks, Richard. One of the things that you were talking about, Stephen, earlier was about the cognitive impact of the operation. How do you go about preparing a patient ahead of the operation for the fact that they may lose some of their cells? I mean, it was interesting you kind of like showing the pictures and the app and that kind of stuff beforehand. But that must be a very difficult kind of human aspect of the role that you do.
It is, and part of the research we're doing is really to understand this whole process a bit better, because before we didn't test for it, so we didn't really know what effect it would have on people. We just had what patients would report back to us and say what they were noticing. Now we have this data, we can actually use it and try to understand what happens with patients. Interestingly, our initial work we're doing looking at the impact of surgery actually suggests that with patients with their glioblastomas, they come to us with deficits, and they get a bit worse following surgery, and then they all recover back to baseline. So actually, much of the issue with these patients, it happens before we ever see them. That also means two things. First of all, it means that I think rehabilitation is going to be the way forwards for this area. Going to be absolutely essential that we have a targeted way of doing radiotherapy. So very similar, like what Richard's describing in terms of just looking at different gene abnormalities within these tumors, actually looking at specific deficits in patients. And we have a trial that we're just opening at the moment that is looking specifically at emotional recognition problems and how we can train patients out of this. And the second, so that's one aspect of it. The second thing about it is that we can then use this information to inform patients about what's going on, and this is a really important part of it. So if you have this, and you've got data saying, look, these are the problems you're going to have with this, and we know this from other areas, that this will makes a big difference. And I think that's an area that we do need to spend a bit more time doing. Again, it's the thing about having psychologists who can explain this and provide workarounds or give them some. So, for example, patients who've got memory problems, about having notepads near them and making lists and do that to help them as part of that. It's something we do need to look into in more detail.
It might seem a strange question, but how did you both get involved in brains? To put it to simplify, what was the aspect? What drew you to this area of work?
Shall I go first? I, as a very young medical student, doing sort of the preclinical sciences, I really got an interest in the brain and the nervous system. And that really carried on through my clinical training. And every time I did something involving the brain, I really sort of enjoyed it. And then I sort of got into the surgery side, because originally I was thinking perhaps neurology. Then I got into the surgery side of things. It sort of fitted together very well. And I did my first post in neurosurgery some years ago in Nottingham, and thought this is exactly what I want to do. And I really loved it. How I got into neuro-oncology, and this is true, is I went to see the then Professor Pickard, and said, Professor Pickard, I'd like to do neurosurgery and want to do some research. And he said, well, what about doing some research into brain tumours? And literally was that. And I then got into it from that way. And I keep saying to people, I'm still effectively doing the same project he set me back in about 20 years ago. And I still haven't finished that project we started.
Thank you, Stephen.
I don't have an answer as good as that one, I'm afraid. I liked tinkering around in surgically. I found it very interesting, and I think there's something to be said for working both scientifically and manually on the most complex thing in the known universe on a weekly basis. And I probably would say that cancer's arguably the most complex disease that we know about as well. So perhaps it speaks to both of those things.
I think that's an equally good answer, Richard. That's great. One of the questions that's come in again, someone said, are there any of the innovative ways of getting drugs across the blood/brain barrier to treat brain tumours using novel vectors like Zika virus working? Hopefully that makes sense to you as a question.
I think one of my colleagues needs to answer that question. Stephen, can you I might have a slight
So the answer to that question is there are lots of things being looked into that. So there is a big EPSRC grant going through engineering, chemistry, and the nanoscience centre, as well as some of the clinical departments, looking at how you can go about treating these difficult to get at tumours, and brain is one of them they're actually looking at. And there are ways of looking at it by developing polymers that you can put into the cavity of the tumour at the end of surgery that lets these drugs sort of leak out over a period of time. There's others where they're looking at nanotechnology, so looking at bringing that sort of nanoparticles that can be taken up directly into the tumour, and others that are looking at using putting electrodes in that sort of incorporate the tissue, so pushes it into the tissue, and there's some very exciting data coming out from those at the moment, where that might be able to come through. The thing about Zika virus is that there was a lot of interest in Zika virus a couple of years ago, because Zika attacks the stem cells that make develop the brain, and they're the same cells that cause the tumors to keep on replenishing tumor cells. And one of our colleagues has actually looked at this in more detail, and it has basically found that although for some reason he doesn't understand, we're beginning to understand, it doesn't seem to get into these tumor cells as well as we would have hoped. So again, there's a bit of research going on to understand that a bit better. Yeah,
I mean, essentially the microenvironment is hostile and stops it. So, I mean, I think in a way, new therapies are obviously very much needed. But I think that our approach perhaps is to go back and just look sort of more exhaustively at what's currently available and saying, are we really optimizing that? Whether it's from a surgical perspective, whether it's from a radiotherapy perspective, whether it's from a targeted medication perspective, are we throwing out stuff that would actually be useful in certain groups of patients? And so I think that whilst, as I say, our two objectives, one of which is to try and develop treatments for patients of the future, and that's really important. The discovery science is absolutely paramount to that. To help patients that Stephen and I will see in clinic every week, a much better approach, I think, is to really make sure that stuff we've decided doesn't work, like precision therapy or surgery, it's all bound to fail. Well, it might, but actually we all know that in clinical trials, there are those 3 or 4 patients who do really, really well. And can we identify through better characterization and understanding of the molecular neuro-oncology, why that's the case, and then use that to plan treatments more appropriately.
Thank you. And one of the other questions that's come in is, can these techniques be applied to early diagnosis of brain cancer? Sorry, it might be a bit missing out here, as stratification of cancer type. I think I might be missing a word out there, but hopefully
early detection of brain cancer is a bit of a contentious field. So in order to have sort of like a screening test, there are various criteria that you need to fulfill, one of which is good treatments for early disease. But unfortunately, it becomes a little bit chicken and egg because we often don't pick up this disease early. And so we assume that patients do badly irrespective of when we pick the disease up. What we don't want to do with a screening test is identify a disease that is inevitably going to be fatal very rapidly in a patient who would have lived another 6 months without that knowledge. And, so we essentially what's called increase morbidity without improving mortality. That said, I think that's probably a little bit fatalistic, and actually that there is some evidence that if you treat glioblastoma early, patients will live longer. Whether that's their natural disease just playing out, we don't yet know because we haven't got enough information. But in terms of the actual, science behind early detection, there's no reason why things like cell-free DNA and other, biomarker tests, various different metabolic approaches that have been taken, wouldn't work in the early detection field. The issue is that of sensitivity and specificity. So how good are they at picking it up? And if they pick up abnormal signal, how accurate is it that that's a problem that's really there? So there's a lot of work ongoing in that direction, but there are probably lower hanging fruit that will help that before we move it in that direction, if that makes sense. So things like picking up disease recurrence in patients with known glioblastoma that's been treated is a sort of avenue that we're exploring at the moment as a way of saying, look, how low can we go with the signal in these various biomarkers, potentially with the view of then moving that out to some sort of, early detection. But I think we've got to be cautious that we don't just detect things earlier that we still can't do anything about, because that's not going to be great for anyone.
Thank you. I alluded to it earlier, but how? I assume you will be both involved in the new cancer hospital once it's built. How will that change things for you? Will it make the research easier? Will it make trials? What difference will it make for you in both your roles?
I think it's providing the space to have the research and the clinical next to each other. I think is going to be absolutely essential. I think it's going to be really important for a whole variety of different cancers. I think brain is certainly one of them, to have that and have the and also I think it will be a focus that will actually attract some of some of the best and brightest to help further develop our approach on these tumors. I think it's going to be, from that perspective, it's going to be very good to sort of promote Cambridge as a real big centre for people to think about, instead of thinking like the Marsden, which is a big cancer hospital in its own right, thinking of Cambridge itself.
Yeah, no, I'd echo that.
Great, thank you, Bill. And with that, an hour has gone already. It's fantastic that you have. Thank you both for the time that you have given, and to everyone for joining us and who will watch this again. News on the next CBC on Tour event will be shared via our social media channels very soon. But But in the meantime, thank you both again, Stephen and Richard, and hope you all